Thank you, everyone. Let me start by thanking Arnav and Pinaki for giving this wonderful opportunity to discuss some of our results. And I am Shiva from IIT Kanpur. So before I'm going into the topic of discussion today, what I'll possibly do is so I'll take one slide to introduce broadly what do we do in our lab. So I'm an experimentalist, and we broadly focus on two aspects in our lab. One of the aspect is polymers effectively with a specification towards non-equilibrium conditions. We kind of prepare the system, drive the system to out of equilibrium situations. We quench them, and then we try and study them towards their towards equilibrium and stuff like that. And by doing that, our focus effectively is in controlling structure formations and different kinds of instabilities. And also, once we quench it, one ways of getting that idea is to kind of get focus on residual stresses and stuff like that. What we are trying to look at is the molecular reasoning underlying this kind of residual stresses, which is one aspect that we work on. And the another aspect is also we kind of think on understanding the dynamics of bacterial communities, both at large density bacterial suspensions, trying to understand their collective dynamics, looking at aspects of turbulence, and where we recently have shown that that in contrast to conventional turbulence, bacterial turbulence do show very strong presence of Levi walk until the fluid is correlated and stuff like that. And also we kind of do a very close collaboration with biologists, where we kind of try to understand how stresses play a significant role in switching the lifestyles of bacteria and stuff like that, right? So with that, which is not what I'm going to discuss today, and what we'll be discussing today is some recent aspects which is not yet published, which we are summarizing the results, trying to understand a simple deposition of particles, and instead of depositing passive particles, we are replacing the passive particles with those particles which can now swim and come and attach to the surface, and may not absorb with the full probability, may kind of diffuse through and then get absorbed kind of a little longer. So by just introducing this degree of freedom, so how does results change, and the kind of surprises that what we have seen. And of course it's important that I highlight that this is from a lot of hard work from these two graduate students from my group. This project is led by Hemlata, and Dhananjay is also sitting here in the audience. And as I said, we do a lot of collaboration with these biology guys, and we get the bacteria from them, and that's Saravan from biology department, and thanks to all the funders for funding our activities. And yeah, so let's start. When I think about interfaces, growing interfaces is something that we see in various aspects, right? On left, what I show you here is a Belousov-Zabotinsky reaction, chemical oscillations which are growing, and then on the right, you might have seen the movie, these are turbulent liquid crystals where the different phases kind of grow into the other phases. And people have seen ink imbibition and paper combustion and stuff like that. Interfaces which are growing is something that we all have seen in one of the other forms. And also in the living systems, there are a lot of very interesting aspects. On left, what I show you here is a dendritic swarming colony of a bacteria, which is an interface which is growing, and this is at a much larger scale. At the local scale, the bacteria is still swarming, and you see a lot of collective dynamics, and which drives such patterns. And also people have shown with monkey cells, on left what I show you is monkey cells where there's interfaces which are growing and so on. And just to give some background, especially for the younger colleagues here, to give some aspect of how do we think about deposition of particles, right? So just think about particles which are coming and settling down, and if one wants to write a simple continuum equation with respect to that, so we are thinking about an height which is growing in time and which is kind of getting smoothened like a surface tension term that you are having, and then you have a stochastic noise in here, and then this is an equation which one can analytically solve. And if one does this for a one-dimensional system with another one being time, one gets the roughness of this interface scaling with the system's size to the power of, and the roughness with time to the power 1/4. Of course, this is at much later times when the system has kind of reached saturation and stuff like that. And then another thing which the famous Kardar-Parsi-Zang's inclusion, which is if one thinks locally, there are nonlinear corrections one has to think about for the flux field, and if one does that, that changes the exponents, both of this roughness and growth exponents, right? And just to summarize, effectively what we are thinking about are interfaces which often follow power laws, where the roughness scales with the size of the system, or in the Fourier space it scales with the wave vector, with is also again one can get the roughness exponent in both the ways, or one can think about the time evolution of the roughness, or if since the flux is linear, constant, one can also replace time with the mean height of the interface and stuff like that people have been showing in different experiments, right? And then thanks to Vichak family scaling, one also now knows that the relation between alpha and beta, while the time correlations come in at smaller times, at later times when you see a saturation with time, then the length scale correlations becomes much more evident, and the relation between alpha and beta are captured by what is called as this dynamic exponent. Is that right? So that's all about the background, and given this background, what we thought about this, we have discussed about the ideas of this, and there are papers which are published which is saying that one can essentially experimentally capture this idea and one can map it on to KPC type of interfaces and stuff like that. And what if I have now an interface which is formed, and now as you can see that it's not just that it is kind of adducted towards the edge only, that they can now also do some random motion. If I think about an interface which is forming like this and that it is growing, what kind of surprises or what kind of aspect that one can think about, right? And the focus of today's talk is trying to understand, set up this kind of interface, and then look at the results both from the experimental perspective and a very simplified description of model, right? Okay, so yeah, first we want to create interfaces, and creating interfaces is not a straightforward task, and we just used a very simple idea. We are all used to stain formation of a coffee drop when it fall on our shirts, on our tables and stuff like that. And if you see the coffee drop, you see the edge is much more thicker, which is the coffee particles are much larger in terms of concentration at the edge. And why is this happening? If one thinks about it, just put in a drop, and then because of the geometry, the evaporation at the edge is going to be larger, and now just to accommodate space, you lose lot of volume here, but then this is energetically much more costly, which means to have surface energy minimization, one has to have an advection of the bulk fluid from the center of the drop towards the edge of the drop. Now assume the same thing with particles. Now if you have the particles, while the center fluid is getting advected, it also drags the particles towards the edges. So now if I sit on here and then look from the top surface, I am seeing an interface which is being formed, right? So that's the idea that we are going to look into. And then, as we promised, the first thing is we want to compare the idea with active system and a passive system. So for that, what we chose, of course, geometry is important, so I cannot just replace living system with some other shaped static passive particles. So that's why we chose two different bacterial systems. One is Bacillus subtilis, which is a very strongly swimming entity in our case, and E. coli, whose swimming is strongly repressed. Right, and now what I'm showing you here is the trajectories which are averaged over I think few seconds, and the color maps indicate the velocities, and here I'm showing you the distribution of the velocities, you can speed effectively, and you can see that those which are swimming effectively have a swimming velocity of the order of 20-30 micrometer per second, and the guys which are not swimming are kind of most localized, and if you look at the velocities, this is more than an order of magnitude small. So effectively one can think about an interface which is forming with Bacillus and an interface which is forming with E. coli, and try to find out what kind of interfaces we have. Right, so that's a video, I think a little accelerated, 8 times faster than the real timings of the experiments. You can see, let me rerun the video. Right, so now the passive guys more or less get advected because of this advection of the fluid flow, while the swimmers, you can see like they are coming and forming this interface. There's a finite interface which is forming, but it is not that like, it is like kind of coming and just attaching. They also can move around, right? And now, if one thinks about again the velocities of the advection and the swimming, what is interesting is that even though these guys are not moving much, the advection flow is much stronger. That like the velocity to which they are coming and forming the interface, the flux is very similar in both the cases. So that there is no difference between the active and passive systems in terms of the flux, right? And with that, now we have to characterize the interface, and now what I'm showing you here is like fast forwarded video with the red line which is effectively capturing the interface. The one bacteria comes and falling, right? And once one focuses it, like as Silke has also highlighted, that one can see certainly overhangs which are visible, and hence defining H as a single valued function becomes problematic. But then like there are earlier works like including a review by Takeuchi, where one can show that like if the overhangs are not very strong, one can in principle neglect that and define whatever the maximum height that one has as the single valued height function. And while not just kind of inspired by the literature, we also thought like, okay, let's just do our own system. Like now what I'm showing you here is that like the actual interface which is shown in the black squares, and now like the red lines are effectively taking the height at every location, whatever the maximum height is. So I'm neglecting overhangs in one case and including overhangs in another case, right? And then we did just a simple box counting method and try to find out what the dimensions of this fractal object what we are having, and one can see that like they're coming out to be pretty closer, and we have done it for different times and different species, and it's not changing much, and hence neglecting overhangs is not a very serious problem in our case, I presume, and that's how we go ahead. So I define height as the maximum at a given location, and as a function of time this evolves, and then to support this analysis, I also parallelly show the factor analysis where I don't neglect the overhangs, right? So that's essentially the background. So now for that, of course, I define the basic statistical parameters. One gets h at every x location as a function of time, and you define the mean height and the roughness around the mean height, and that gives you the two statistical characteristics then one looks into. As I started introducing, what we are interested in is in correlating evolution of roughness with time, or in other words, correlating roughness with mean height. In experiments, finding zero time is little difficult, and that's why we usually rely on correlating roughness and mean height, right? Okay, so that's what I'm showing you here. First results, and this you have seen already. And now if I plot the roughness as a function of mean height, phi m defines the fraction of the swimmers. So phi m0 is basically a passive particle, passive bacteria E. coli, and phi m equal to 1 is like all of them are swimming, is a Bacillus subtilis, and you can see like by looking at this evolution, the exponents for the swimmer and the non-swimmers are strongly different, right? And just to give you some, so this ash color symbols what you are having is the actual experimental data, which is collected like every 30 frames per second, and then this on top what you are seeing is blue symbols are every 3 seconds I'm putting in one symbol, just to show like that it's not much is varying in terms of the time there. Right. So then if really wanted to identify if such transition really exists, what would be like ideal is to one think about systems which has different activities, or one think about populations like which has like different populations of active system in a suspension, right? So how do we do that? We cannot just mix E. coli and Bacillus, because biologically they create other issues, and that's why what we decided is like, okay, this guy is swimming really fast, and let's just now remove the nutrients from them and controllably remove the nutrients, so that you are playing with the metabolic activities of the system. So you keep waiting, so then like slowly they start losing their activity, right? And that's what I show you here. Sorry?
Passive, right?
Yes.
So that's not 1/3?
It's not 1/3. For instance, experiment similar to this was performed in Arjun Yacht's group with rod-like particles. No, rod-like particles show kind of quench KPC. So they show that like the beta is of the order of 0.67, 0.7, and their alpha is also comparably different. So it's not KPC class, right? Yeah. Okay. So now what I'm showing you here is like, I took the swimmers, removed the nutrient at time t equal to 0. This is how they are swimming, and I just waited for a few hours, and you can see like, while the density, the number of swimmers are very similar, so one can still think that like the particle number is conserved throughout this, and they are not multiplying because of the reduced metabolic activity, and one can see that like here all of them are swimming. Here few of them are swimming and few of them are like more or less static, as though like you kind of separate a population of guys which are swimming and guys which are not swimming. So then we looked into the distribution of this speed, and if you just see t equal to 0 minute, you can see this long tail which is coming, and with a peak at lower value, and with time you can see the peak at the lower value keeps increasing and the tail keeps shrinking. So effectively the population which is swimming is kind of decreasing, and the population which is non-swimming is increasing. So now based on this idea, I can effectively integrate out the area of this regions, and I can define a fraction, motile fraction, as the ratio of these two areas, right? And that's how we defined the motile fraction, and once we define and then get it for all the systems that we have done, and you can see that like this is our pure swimmer. Now we can go from something like 0.7 percentage, 0.7 fraction to something like 0.1 and then 0, which we had this non-swimmer, two extreme cases, and that now we can control in between, right? And now let me show the first results with this mixed population. You can see like phi m 0.68, which is 68% of them are swimmers and the remaining are non-swimmers, and down to phi m 0.1, which is 90% of them are non-swimmers and 10% is swimmers. And you can see while the guys which are swimmers display very similar exponent as what we have seen for the pure active system, the ones in between, like the mixed value, like almost 50% of mixtures of swimmers and non-swimmer show a lesser exponent, which means that like the kind of roughening is like a smoothing is more efficient at an optimal fraction in between, right? And just we'll go ahead and show you little more results on that. We just wanted to also look at other characteristic exponents. For instance, we were looking at the roughness exponents, and none of this activity seems to really change the exponent here, and which indicates alpha is of the order of 0.45, and within the exponent one can say it's 0.5 or something like that. And then we also did this without neglecting the overhangs, looked at the fractal dimension calculation. The fractal dimension seems constant, and we have done for a series of systems here. And now beta, which is the growth exponent, is now plotted as a function of phi m. Alpha and df is also plotted as a function of phi m. Both alpha and d are not changing with phi m, which is more or less constant. And the surprise comes when beta seems to have a non-monotonous behavior with phi m. For purely active systems, completely swimmers, we had something like 0.5, and in between it kind of goes via this 0.3-ish. I don't know if I can call this as KPC, but I'm refraining myself from not calling it with anything. So this is just some decrease in the growth exponent values and then increases again. Yes, Satya. I don't think so. I don't think Pichek scaling will hold, because now your alpha and beta are supposed to be coupled. Now we see that alpha and beta are decoupled, right?
Z itself changes.
Could be. So that's what, if one just takes a ratio, it would indicate that is changing. But I don't know if I would rely on family Pichek kind of scaling to rescale things and stuff like that. Right? Yeah. So that's one. Such decoupling between beta and alpha is not predicted, and also this fractal dimension being constant is something one would have expected with alpha being constant, because that alpha and the fractal dimension are related to each other. And so we started asking, okay, these are all at a interface level, and we don't know why this is happening. So maybe we will find some clues of looking at the process of how this interface is forming, right? So let me take you through what we think could possibly happen. right? So now we started with a sessile drop, right? And now we are thinking about an object which is floating on a sessile drop. And now any object which is floating on a fluid, you know that like it creates this capillary deformation. And now let's say like if this object is moving, right? And now if this movement is more faster than the relaxation of the capillary deformation that what we are having, effectively what they created is a trail of capillary deformation. And now if there are some other bacteria which are coming here and here, so they would rather be attracted towards this trail because of the Laplace pressure, right? So now if that is happening, maybe that could indicate like why we have this very rough like beta being 0.7 or something is happening. Just we looked into experimental clues if this is happening. Now if I just kind of bring your attention towards some of this trajectories which they are coming and forming the interface, you can see that like they kind of switch lanes and like kind of coming and forming at a given location more prominently. This indicates that like somehow they are going towards a given spatial location, which might be the reason behind the larger beta what we are seeing. And then now this is for a passive system. Here activity is not there. This is just the geometry which is playing a role. So now the moment I put in activity, now if you see this swimming guys are like moving around the interface, which means if such quenching lines are existing, so then they would rather randomize the line, and then one would expect that a randomized value is what one would see, and beta being 0.5 seem to indicate that. But what is the reason why we have an optimal fraction in between where the beta is much smaller, that we don't know yet. And now if this smoothening is what is happening, so what we did is like we kind of looked into the height fluctuations, right? So what I'm plotting here is the mean height fluctuation which is ensemble averaged for different phi m's as indicated here. And now our interface is always growing in time, so I have to appropriately normalize with the growth of the mean height, and we are removing that. We are now looking at only the fluctuations. And now if the active particles are coming and smoothing this interface, what would you expect is that like this fluctuation is now not happening as strong as without activity, right? And that's what this gamma is indicating. Now with activity, with increase in phi m, you can see that the slope is decreasing systematically, which says that like all that the guys are doing at a given location is effectively it is trying to smoothen the fluctuations. Now this is at the height level. Now if you think about the particles are now also moving around, right? And now if I look at the mean square displacement of these particles which are right at the interface, now at delta t equal to 0, the particle has just come and stayed as a part of the interface, and then now we are tracking this particle, right? These particles now, if I have a active system, they are like kind of really swimming around, and that exponent indicates that you're really having a like kind of a very almost a ballistic kind of motion. And now if I have like non-swimmers, like passive guys, they are coming, they just rearrange locally, close to being random, and then just get settled completely, right? And now if I think about the mixed population, right, and there we have seen all type of entities. So there are entities which are swimming around because we have a population which is swimming, and then there is a population which is not swimming, and then there is also another population which is kind of trying to swim around, and but then get stuck because there is a region which is growing. They are trying to swim around here, and this is going faster, and then this is just getting stuck, as though you're feeding this growing interface from the sides, right? And that is something that what we have seen. And also while we are looking at this, another thing which came to our attention is that when you look at the interface which is forming, so this is at time t equal to 1, this is with increase in time, a bacteria which is coming and becoming a part of the interface here. So now with time what happens is they kind of jump around and then finally get settled at a different location, right? So now this is what I call as this length, the jump length, before they become a part of this interface. And now this jump length, we decided to just look at the distribution of this jump length for different systems, pure swimmers to passive guys. And now the first one, let's go to the passive guys. What is happening is now I'm showing you a rescaled jump length with the length of the bacteria. And you can see all the jump, there is no jump beyond the length scale of the bacteria itself. So they come, they just rearrange, and then they just get stuck. That's what is happening with the passive system. And for the active system, if you now focus on this black square symbols, you can see that more or less for almost up to 10 times the bacterial length, all of them kind of move around before they can get stuck, right? And then it starts decaying much more strongly, and in mixed populations we have a slope which is different from the passive population. So it has more larger jump length distribution, right? So this we will come back to little later. Before I'm going further, let me just also think why we are seeing alpha constant, right? So now the way we are doing is like this is the actual interface, and now when I recast into this height variation, which is plotted normalized with the mean height. Now this video is a time video. Now you can see that like there are movements across this interface, which is like kind of reflecting the movement of the bacteria along the interface. And how do we calculate the alpha? We are calculating alpha at the latest stage, so hence swimming is not going to really matter. Even if that happens, we also looked into different times. Now we are kind of like going to get different scales. Like at a small scale you get W from each box and average it out, and at large scale again you average out. So whether the bacteria is being a part of this box or this box, it actually doesn't matter because it is kind of getting averaged out finally, and that's why we think alpha is not changing, right? So now just to summarize, the main thing which seems to happen is that that before the interface is forming, there is a new degree of freedom which was not discussed in earlier deposition of particles, which is effectively you have this jump length distribution. So if one tries to summarize the idea of what experiments indicate, so basically the non-motile guys come in and get adsorbed with a probability 1 and with no jump length probability, and the motile guys coming and more or less they are not adsorbing at all the moment they are seeing, they move around and then they get adsorbed for a finite length distribution, and the mixture will have a contribution from both of these guys, right? And if this is really kind of summarizing the ideas that we were discussing, so then in principle I should be able to set up very simplified model just trying to bring in these ideas into a well-known model, right? So that's what we tried doing. So to start with, we chose like a ballistic deposition model, and where we chose in such a way that there are spurge of particles which are coming and settling, like 800 particles at a given time, which means they can now get attached in whichever phase they are seeing. So we just allowed the overhangs because to mimic similarly what happening in experiments. And so these are sticky particles, so as I said, they can get attached wherever they are seeing the first neighbor, and then we are using a box of length 1000 with 1 being the particle size, and so at a given time we are sending in 800 particles and we are waiting for 10,000 steps and seeing the interface is going, right? So now this is a nominal system, and now we have to bring in the experimental inspirations into this model. So what did we do to think about this geometry induced quenching? There are some regions where they can particularly go and get settled. To mimic that, what we did is when these particles are coming, they also have an extra additional freedom. They can move around for 7 pixels, and if they find any neighbor in any of the 7 pixels, they first get attached there, which means if you have some regions which are growing, they will grow at the expense of the other regions, right? So this so then if that is growing, so then you are having now few locations which are growing more prominently than the other regions. So that kind of try to mimic the quenching that what we are seeing in the experiments, and then we have to also bring in the activity. So the way we did is that now the particles are coming, so now we are for each particle we are giving in a probability on top of that, and this probability is now Pm minus 1 is the probability with which they come just attach, and Pm is same as phi m what we discussed in the experiment. So then they can if you now control this population difference, they can get attached or they can move and stuff like that. So that's the idea behind this model. And with that, now let's first show you the flux that we got. So all the flux is more or less linear for different systems, and now we wanted to do exactly same things what we did in the experiments. Look for the maximum height, look for the roughness, and look for the correlation between the parameters, and that's what I'm showing you here for all the systems. Right. So let me take you through different graphs. So this is a non-motile guy. So now what we said d equal to 7. So they can move around. There is a particular location which can grow. So which means your beta is expected to be larger, and we see beta is close to 0.6, 0.67, and then it saturates at a larger region, and which is expected, Raja length scale. Right. And then, for the motile guy, so they are coming and then they kind of randomizing everything. So there is that probability we just brought in with jump, the probability of jump is 1. They can now move around like from ranging from 20 to 1000 length, and the probability of all of them is equal. Right. So now with that we could get similar to what we were getting from the experiment, which is 0.5. And then for the mixed population, what we see is like initially we still had something similar to 0.66, because the passive guys are like dominating at the initial time scales or the initial mean heights, and then later on like kind of saturates towards getting into 0.33, right? And then this is just like what I summarized. So now betas on this dashed line is what I just shown you in experiment. So effectively this simplified description seems to capture that the variation in beta is coming from the phi m fraction what we had, and then of course we have to also look into whether the alpha is being constant or not, and we looked into the PSD, and that seems that it's more or less constant for the simulations what we have seen. Right. So with that I come to the conclusion, and I hope I conveyed the excitement of our experiments, where controlling the swimmer fraction in experiments and in the simplified model that what we are showing allows us to switch the growth exponents from 0.67 to 0.3 and to 0.5 in a very non-monotonous manner, and such decoupling of growth and roughness exponents are not predictor or expected, and hence I think there is something more to think and discuss about. So with that, thank you.
Thank you. Thank you.
Yeah. So, I mean, in your experiment, I mean, you don't look at really the finite size effect, right? I mean, the family which
The drop size is really larger.
But you could look at the spatial height correlation function. That's another way of getting the Z and alpha and beta things, right? Did you look at that?
We looked into not really a correlation function, but I thought PSD is somehow reflecting that.
So I guess, I mean, I don't know if I understood what you're saying, but I mean, could these, the fact that you're seeing these exponents changing, could it be sort of an artifact of where, in what regime you are? I mean, if there are crossovers,
Right.
For the different phi m's, could you be in different regimes where you're estimating?
Well, so the regime transitions usually is there is a regime where your roughness scales, there is a kinetic roughening, and then it kind of saturates, and then only the other regime really takes over, where your alpha seems to contribute. So in our case, what we have seen is that our roughness is always increasing with the time, with the experimental window what we are seeing. So I think we are always in the same regime, and whatever transitions we are seeing is beyond the regime switches.
Yeah, in the presence of overhangs would be much more in the passive case compared to the active case, right? So, so that could also affect your exponents, because they're not very, very different exponents. Right?
Yeah. So we kind of did the same reference with all the fractions that we have worked with. Overhangs with and without overhangs, we have not seen much variation in terms of the fractal dimension or in terms of the roughness exponent. Right. So now for any time evolution, one has to think about some way of capturing this time evolution. That's why we kind of went ahead with this neglecting the surface, the overhangs. Right. I'm not sure if they are having any differences in terms of phi m. So we have looked into, and we don't see any difference in terms of the extent of overhangs.
Yes. Question, comment in the same direction. So for us, we had trouble with overhangs. And for example, we were naively tried to compute spectra in our system. We were very happy because they looked good, and then realized most of our spectra was overhang noise. So did you have any problems like that?
So when you say
must be quite tricky, yes.
When you say spectra, you mean in spatial?
Fourier transform of our height field in the end.
Yeah, that's what I was showing in terms of neglecting overhangs and remove, with this box counting method. So this being similar, I'm not sure if that is going to be a real reason behind any of our observations.
for us it turned to be much worse than we thought it would be because we have very few actually, but yeah.
No, I agree with you because like that's what we also thought when we started with. But then when we did this analysis, we could clearly see that overhangs are not really playing a dominant role.